We are going to start with the chapter number 6 of class 11th, Anatomy of Flowering Plants. You can very easily see the structural similarities and variations in the external morphology of larger living organisms, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plant is anatomy. What is morphology? Study of external structure. Study of internal structure is anatomy. Plants have cells as the basic unit. Cells are organized into tissues, and in turn the tissues are organized into organs. Different organs in a plant show differences in the internal structure. Within angiosperm, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. Now, moving for the tissue system. We were discussing types of tissue based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. The structure and function would also be dependent on the location. On the basis of the structure and location, there are three types of tissues. These are epidermal tissue system, the ground tissue or fundamental tissue system, and the vascular or conducting tissue system. Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and the hair. Trichomes are multicellular. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single-layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose the stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of the guard cells away from the stomatal pore are thin, and the inner walls towards the stomatal pore, they are highly thickened. Very important line. The guard cells possess chloroplasts and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of guard cell become specialized in their shape and size and are also known as subsidiary cells. The stomatal aperture, guard cell, and the surrounding subsidiary cell that together called what? Stomatal apparatus. The stomatal aperture, guard cells, and surrounding subsidiary cells are together called stomatal apparatus. The cells of the epidermis bear a number of hairs. The root hairs are unicellular elongations of epidermal cells and helps absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. The ground tissue system. All tissues except epidermis and vascular bundle, they constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary layers, in the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. The vascular tissue system. The vascular system consists of several complex tissue, the phloem and xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between the phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. In monocots, the vascular bundles have no cambium present in them. but in dicots they have cambium. Hence, they do not form secondary tissues. They are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in the roots. In conjoint type of vascular bundles, the xylem and phloem are joined situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots and stems and leaves, it is convenient to study the transverse section of mature zones of these organs. Dicotyledonous root. Look at the figure. It shows the transverse section of a sunflower root. The internal tissue organization is as follows. Sunflower root 6.3a. Yes, this is sunflower root. Above here is root here, then epidermis, then cortex, then endodermis, then pericycle, then protoxylem, metaxylem, pith, and phloem. Okay, this is for dicotyledonous root. Yes, tell me is the outermost, maybe later. The outermost layer is epiblema. Many of the cells of epiblema, they protrude in the form of unicellular roots. The cortex consists of several layers of thin-walled parenchymatous cells with intercellular spaces. The innermost layer of cortex is endodermis. It comprises a single layer of barrel-shaped cell without any intercellular spaces. The tangential as well as radial walls of endodermal cells have a deposition of water-impermeable waxy material suberin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in the cells. Pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and phloem, they are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later a cambium ring develops between xylem and phloem. All the tissues on the underside of the endodermis such as pericycle, vascular bundles and pith constitute stele. Now monocotyledonous root. The anatomy of monocot root is similar to the dicot in many respects. It has epidermis, cortex, endodermis, pericycle, vascular bundles and pith. As compared to the dicot root which have fewer xylem bundles, there are usually more than 6 xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. Now dicotyledonous stem. This transverse section of a typical young dicotyledonous stem shows the endodermis is the outermost protective layer of the stem. Now head. Covered with the thin layer of cuticle. It may be a trichomes and few stomata. The cells are arranged in multiple layers between epidermis and pericycle. They constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded, thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of cortex is called endodermis. The cells of endodermis, they are rich in starch grains, and the layer is also referred to as a starch sheath. Pericycle is, these are diagrams. Pericycle is present on the inner side of endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. In between the vascular bundles, there are a few layers of radially placed parenchymatous cells which constitute medullary rays. Interfascicular cambium originates from medullary rays. Yes, important question. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is a characteristic of dicot stem. Ring arrangement of vascular bundle. Each vascular bundle is conjoint, open and with enlarged protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by sclerenchymatous bundle sheath. In dicot, it is collenchymatous. Yes, and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. Dorsoventral leaf. The vertical section of dorsoventral leaf through the lamina shows three main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers the upper part, adaxial epidermis, and lower surface abaxial of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between upper and lower epidermis is called mesophyll. Mesophyll, which possesses chloroplast and carry out photosynthesis, is made up of parenchyma. It has two types of cell, the palisade parenchyma and spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically in parallel to each other. The oval or round and closely arranged spongy parenchyma is situated below the palisade cells and extends to lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and midribs. The size of vascular bundle are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick walled bundle sheath cells. Look at these pictures. Yes. Now, isobilateral or monocotyledonous leaf. The anatomy of isobilateral leaf is similar to dorsoventral leaf in many ways. It shows the following characteristic difference. In an isobilateral leaf, the stomata are present on both the surface of the epidermis. The mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along with the veins, they modify themselves into large empty colorless cells. These are called bulliform cells. When bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize the water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundle except in main veins as seen in vertical section of the leaf.